Hello dear students, today the topic of our deliberation is methods of isolation of pure culture and culture characteristics. The study has been categorized into following objectives: to be able to understand pure culture and describe various methods to obtain them. Second, describe bacterial structure in terms of colony, morphology, cell shape and growth patterns. And finally, to be able to differentiate bacteria based on morphology and upon macroscopic examination. First, the students will start with methods of isolation of pure culture. Microorganisms are generally found in nature as mixed populations. To study the specific role played by a specific microorganism in its environment, one must isolate the same in pure culture. A pure culture theoretically contains a single bacterial species. There are a number of procedures available for the isolation of pure cultures from mixed populations. A pure culture may be isolated by the use of specific media with specific chemical or physical agents that allow the enrichment or selection of one organism over another. Pure culture involves not only the isolation of individual microorganisms from a mixed population, but also the maintenance of such individuals and their progenies in artificial media where no other microorganism finds its way to grow. However, it's not easy to isolate the individual microorganisms from the natural habitats and grow them under imposed laboratory conditions. For this, a great deal of laboratory manipulation is required. It is necessary to make the colonies well isolated from each other so that each appears distinct, large, and shows characteristic growth forms. Such colonies may be picked up easily and grown separately for further study. Several methods for obtaining pure culture are in use. Some common methods are in everyday use by a majority of microbiologists, while the others are methods used for special purposes. The first method employed for generation of pure culture is spread plate method. In this method, the mixed culture or microorganism is not diluted in the melted agar medium, unlike the pour plate method. It is rather diluted in a series of tubes containing sterile liquid, usually water or physiological saline. A drop of so diluted liquid from each tube is placed on the center of an agar plate and spread evenly over the surface by means of sterilized bent glass rod. The medium is now incubated. When the colonies develop on the agar medium plates, it's found that there are some plates in which well-isolated colonies grow. This happens as a result of separation of individual microorganisms by spreading over the drop of diluted liquid on the medium of the plate. The isolated colonies are picked up and transferred onto the fresh medium to ensure purity. In contrast to pour plate method, only surface colonies develop in this method and the microorganisms are not required to withstand the temperatures of the melted agar medium. Second technique for generating pure culture is streak plate method. This method is used most commonly to isolate pure cultures of bacteria. A small amount of mixed culture is placed on the tip of an inoculum loop or a needle and is streaked across the surface of the agar medium. The successive streaks thin out the inoculum sufficiently and the microorganisms are separated from each other. It is usually advisable to streak out a second plate by the same loop or a needle without re-inoculation. These plates are incubated to allow the growth of colonies. The key principle of this method is that by streaking, a dilution gradient is established across the face of the Petri plate as bacterial cells are deposited on the agar surface. Because of this dilution gradient, confluent growth does not take place on the part of the medium where few bacterial cells are deposited. Presumably, each colony is the progeny of a single microbial cell, thus representing a clone of pure culture. Such isolated colonies are picked up separately using sterile inoculating loop or a needle and re-streaked onto fresh media to ensure purity. The third important technique employed to produce pure culture is pour plate method. This method involves plating of diluted samples mixed with melted agar medium. The main principle is to dilute the inoculum in successive tubes containing liquefied agar medium so as to permit a thorough distribution of bacterial cells within the medium. Here, the mixed culture of bacteria is diluted directly in tubes containing melted agar medium maintained in the liquid state at a temperature of 42 to 45 degrees Celsius. The bacteria and the melted medium are mixed well. The contents of each tube are poured into separate petri plates, allowed to solidify, and then incubated. When bacterial colonies develop, one finds that isolated colonies develop both within the agar medium and on the medium. These isolated colonies are then picked up by the inoculating loop and streaked onto another petri plate to ensure purity. Pour plate method has certain disadvantages as follows. First one is the picking up of subsurface colonies needs digging them out of the agar medium, thus interfering with other colonies. And the microbes being isolated must be able to withstand temporary exposure to 42 to 45 degree temperature of the liquid agar medium. Therefore, this technique proves unsuitable for the isolation of psychrophilic microorganisms. However, the pour plate method, in addition to its use in isolating pure cultures, is also used for determining the number of viable bacterial cells present in a culture. Another important method is serial dilution method. As stated earlier, this method is commonly used to obtain pure cultures of those microorganisms that have not yet been successfully cultivated on solid media and grow only in liquid media. A microorganism that predominates in a mixed culture can be isolated in pure form by a series of dilutions. The inoculum is subjected to serial dilution in a sterile liquid medium, and a large number of tubes of sterile liquid medium are inoculated with aliquots of each successive dilution. The aim of this dilution is to inoculate a series of tubes with a microbial suspension so dilute that there are some tubes showing growth of only one individual microbe. For convenience, suppose we have a culture containing 10 ml of liquid medium containing 1000 microorganisms, that is 100 microorganisms per ml of liquid medium. If we take out 1 ml of this medium and mixed it with 9 ml of fresh sterile liquid medium, we would then have 100 microorganisms in 10 ml or 10 microorganisms in 1 ml. If we add 1 ml of the suspension to another 9 ml of fresh sterile liquid medium, each ml would now contain a single microorganism. If this tube shows any microbial growth, there is a very high probability that this growth has resulted from the introduction of a single microorganism in the medium and represents the pure culture of that microorganism. Yet another important technique of isolating pure culture is single cell isolation method. An individual cell of the required kind is picked up by this method from the mixed culture and is permitted to grow. The following two methods are in use. First one is capillary pipette method. Several small drops of a suitably diluted culture medium are put on a sterile glass cover slip by a sterile pipette drawn to a capillary. One then examines each drop under the microscope until one finds such a drop which contains only one microorganism. This drop is removed with a sterile capillary pipette to fresh medium. The individual microorganism present in the drop starts multiplying to yield a pure culture. The another method is micromanipulator method. Micromanipulators have been built which permit one to pick out a single cell from a mixed culture. This instrument is used in conjunction with a microscope to pick a single cell, particularly a bacterial cell, from a hanging drop preparation. The micromanipulator has micrometer adjustments by means of which its micropipette can be moved right and left, forward and backward, and up and down. A series of hanging drops of a diluted culture are placed on a special sterile cover slip by a micropipette. Now a hanging drop is searched which contains only a single microorganism cell. This cell is drawn into the micropipette by gentle suction and then transferred to a large drop of sterile medium on another sterile cover slip. When the number of cells increases in that drop as a result of multiplication, the drop is transferred to a culture tube having suitable medium. This yields a pure culture of the required microorganism. The advantages of this method are that one can be reasonably sure that the cultures come from a single cell and one can obtain strains within the species. The disadvantages are that the equipment is expensive, its manipulation is very tedious, and it requires a skilled operator. This is the reason why this method is reserved for use in highly specialized studies. Finally, the enrichment culture method. Generally, it's used to isolate those microorganisms which are present in relatively small numbers or that have slow growth rates compared to the other species present in the mixed culture. The enrichment culture strategy provides a specially designed cultural environment by incorporating a specific nutrient in the medium and by modifying the physical conditions of the incubation. The medium of known composition and specific conditions of incubation favors the growth of desired microorganisms, but is unsuitable for the growth of other types of microorganisms. Now, dear students, we'll discuss the second objective of the study, which is culture characteristics. When a single bacterial cell is deposited on a solid or a liquid medium, it begins to divide. These cells divide exponentially, eventually forming a colony. When grown on a variety of media, microorganisms will exhibit visible physical differences in appearance, in their isolated colonies, and in their growth. These differences are called cultural characteristics or morphology. Cultural characteristics may be used as an aid in identifying and classifying some organisms. These physical characteristics are often specific for the type of bacteria and can be used as a means of recognition. The appearance of colonial growth on agar medium can be very distinctive for individual species. Some microorganisms have characteristic growth patterns, but they aid in the identification of species only if they are distinctive. Although some bacteria grow in distinctive patterns, others look alike. Colonial morphology, however, is influenced by the media and other growth conditions. The colonial morphology of the same bacteria may vary on different media or under different conditions. Cultural characteristics or morphology are determined by culturing microorganisms on nutrient agar plates, in nutrient broth, and in slants. After incubation, the characteristics are observed. Observation of such factors also helps in recognizing types of bacteria. Differences in the macroscopic appearance of a microorganism's growth are used as a basis for separating microbes into taxonomic groups contained in Burgess' Manual of Systematic Bacteriology. Now, I briefly rush through the characteristics of microorganisms in solid nutrient agar. The first important characteristic is size. It is a cultural characteristic in nutrient agar plates. Pinpoint, small, moderate, or large. Different types of bacteria produce different sizes of colonies, from the punctiform or pinpoint colonies of Streptococcus pyogenes and Staphylococcus epidermis to the large colonies of Klebsiella pneumoniae. The size of a colony can give some important clues as to the identity of the organism under observation. Second is pigmentation, a cultural characteristic in neutral agar plates, that is, color of colony. Some bacterial cells are pigmented, as in some of Micrococcus luteus and Chromobacterium violaceum. Some bacteria like Pseudomonas aeruginosa produce a water-soluble pigment that diffuses into the agar. Next is form or the shape, a cultural characteristic in nutrient agar plates. The shape of a colony as described as follows: punctiform, a type of form in nutrient agar plates. Colonies which are very small but not microscopic have a diameter of less than 1 mm. Circular, a type of form in nutrient agar plates. Unbroken peripheral edge. Filamentous, a type of form in nutrient agar plates wherein colonies are composed of long, interwoven, irregularly disposed threads. Next is irregular, a type of form in nutrient agar plates, indented peripheral edge. Rhizoid, a type of form in nutrient agar plates with root-like spreading growth. Spindle, a type of form in nutrient agar plates, spindle-like, bulged at the middle while pointed at the ends. Next is elevation, a cultural characteristic in nutrient agar plates. The degree to which the colony growth is raised on the agar surface is described as flat, raised, convex, or ambonate. Flat, a type of elevation in nutrient agar plates. Elevation is not discernible. Raised, a type of elevation in nutrient agar plates that is slightly elevated. Convex, a type of elevation in nutrient agar plates which is dome-shaped elevation. Pulvinate, a type of elevation in nutrient agar plates which is cushion-shaped or swelled. Ambonate, a type of elevation in nutrient agar plates which can be raised with elevated convex central region. Margin, a cultural characteristic in nutrient agar plates, the appearance of the outer edge of the colony described as entire, lobate, undulate, serrate, and filamentous. Entire smooth, a type of margin in nutrient agar plates which is sharply defined even at margins. Undulate, a type of margin in nutrient agar plates with wavy indentations. Lobate, a type of margin in nutrient agar plates marked by indentations. Erose or irregular, a type of margin in nutrient agar plates, irregular at margins, tooth-like appearance. Filamentous, a type of margin in nutrient agar plates which can be thread-like, spreading edge. Curled, a type of margin in nutrient agar plates with layered curls. Now the characteristics in nutrient broth cultures. Uniform fine turbidity, finely dispersed growth throughout. Sediment, concentration of growth at the bottom of the broth culture may be granular, flaky, or flocculent. A ring, a film of growth clings to the test tube in the form of a ring at the liquid-air interface requiring oxygen. Pellicle, thick pad-like growth on the surface. Flocculent, flaky aggregates dispersed throughout. Membranous, growth is as a thin skin at the surface. Now the characteristics in nutrient gelatin cultures. Firstly with crateriform, liquefied surface area is saucer-shaped. Napiform with bulbous-shaped liquefaction at the surface. Infusibuliform, a cultural characteristic of nutrient gelatin, funnel-shaped. Now succate, a cultural characteristic of nutrient gelatin with elongated tubular. Now stratiform, complete liquefaction of the upper hull of the medium. Now characteristics in nutrient agar slants. Abundance of growth. The amount of growth is designated as non-slight, moderate, or large. Pigmentation. The coloration of organisms are seen in surface colonies. Most organisms will lack chromogenesis, that is pigment production, exhibiting a white, beige, or gray growth. Pigmentation within the organism may be red, yellow, violet, or other colors. Soluble pigments may be blue, green, yellow, brown, or other colors. Pigmentation occurring within the organism itself or water-soluble pigment that diffuses into the surrounding medium. Optical characteristic, evaluated on the basis of amount of light transmitted through the growth, described as opaque, translucent, or transparent. Next is form or the shape. The appearance of the single line streak of growth on the agar surface is designated as filiform, ecannulate, beaded, effuse, arborescent, rhizoid. First we'll start with filiform, which can be continuous, thread-like growth with smooth edges. Now ecannulate, which can be continuous, thread-like growth with irregular edges. Beaded, which can be non-confluent to semi-confluent colonies. Effuse, ranging from thin spreading growth. Arborescent, which can be thin or tree-like growth. Rhizoid, a type of form to describe nutrient agar slants, root-like growth. Now the consistency. A cultural characteristic in nutrient agar slants can be described as dry, buttery, or mucoid. Dry, a type of consistency in nutrient agar slants free from moisture, for example, Bacillus subtilis. Buttery, a type of consistency in nutrient agar slants moist and shiny. Mucoid, a type of consistency in nutrient agar slants slimy and glistening, for example, Klebsiella pneumonia. Dear students, this was all about today's topic, that is isolation of pure cultures and culture characteristics. Hope you enjoyed today's deliberation. Goodbye.